Welcome to Infection and Immunity Evidence Explained, a Doherty Institute podcast. I'm Rebecca Elliott. From protecting local communities to safeguarding global health, vaccines have been transforming public health for over 200 years. According to the World Health Organization, between 3.5 and 5 million lives are saved every year thanks to vaccines, which help protect us from diseases such as diphtheria, tetanus, pertussis, influenza, and measles. Vaccination Through the Ages is the title of the Miegunyah Distinguished Fellowship Public Lecture, which will be delivered by Professor Arup Chakraborty, a luminary from the Massachusetts Institute of Technology, on Monday the 25th of March, here at the Doherty Institute. Today I'm joined by Arup to dive into the fascinating world of vaccines, from their historical roots to the cutting-edge science shaping our future. Arup is a professor of chemical engineering, physics and chemistry at MIT and holds the prestigious title of Institute Professor, an honour only bestowed upon 12 faculty members. He is among the select few who hold membership in all three branches of the US National Academies: Engineering, Science and Medicine. For over 2 decades, Arup's work has focused on bringing together approaches from statistical physics, immunology and virology. His interests span T-cell signalling, development of the T-cell repertoire, and a mechanistic understanding of virus evolution, antibody responses, and vaccine design. Arup, thank you so much for joining me on Infection and Immunity Evidence Explained.
Thank you for having me.
Let's start with the very basics. How do vaccines work?
We have an adaptive immune system, which is remarkable in that it enables us to mount pathogen-specific responses against the vast and diverse and evolving world of microbes. And I'll first give you a sort of 30,000-foot view of how this works. And then if you want to dig deeper, we can do that. So T cells and B cells are key players of the adaptive immune system. And they are capable of mounting specific responses directed toward the protein components of a particular pathogen. And the mechanism by which they do that is quite remarkable. Now, once they've mounted these bespoke responses for a particular pathogen, after they can clear the pathogen, a memory of this event is imprinted in your immune system, and which enables you to mount robust and rapid responses upon reinfection with the same, or reinfection or re-exposure to the same pathogen. And this pathogen-specific immunological memory is the basis for vaccination. So with a vaccine, you try to elicit or activate memory B cells, T cells, and the products of B cells, antibodies that are specific for the bug against which you wish to protect us. So that's the basic concept of how vaccination works.
It's truly remarkable, and we might get into a few of the types of vaccine a little bit later on. But the first vaccine was developed in the 18th century for smallpox. Can you tell me a little bit about the first vaccine?
I'm going to push back a little about your statement that the first vaccine against smallpox was developed in the 18th century. Actually, human ingenuity has been trying to protect people from smallpox for much longer than that. So perhaps as early as about 1500 AD, the Chinese were using a procedure to try and protect people. And what they were doing was they would collect material from smallpox scabs, store it for a while, dry it, powder it, and then administer it to healthy people through a silver tube. And for reasons I don't know, apparently left nostril for women and right for males. And the idea was that this person certainly would develop symptoms and so on, but would often be protected. And the Europeans found that the Indians were using a very similar procedure in the 17th century, with big differences in the way to administer it. And this procedure came to be known as variolation and was brought to the West also later by a remarkable woman called Mary Wortley Montagu. But you see, this procedure of variolation was sometimes effective, but it was very traumatic, its administration and the symptoms, and often was very dangerous because sometimes it would kill the person, and sometimes it would even seed little outbreaks or epidemics of smallpox. So it didn't really firmly take hold in any way whatsoever. But they were doing something that was akin to vaccination. And Jenner came along, and the one you referred to in the 18th century, and so cowpox is a disease that afflicts largely milkmaids when they when they milk cow in those days when they used to milk cows by hand. It used transferred to them from the nipples of the cows who used to get cowpox. That was harmless for humans, more or less. And Jenner heard from a milkmaid that she was actually variolated by this old procedure, and she did not develop symptoms because she had cowpox. And he heard other related stories like this. And so, after much research, he decided to do an experiment, and he actually used the same variolation procedure, but not with material taken from smallpox scabs, but from cowpox scabs that was taken from a milkmaid called Sarah Nims, and the cow that infected her was called Blossom. We know so much about all
Is that
this
a true story?
yeah, we know so much about all this because it was a momentous event,
Yeah.
And,
Blossom the cow.
And so anyway, in May of 1796, he did that to a boy called Phipps, and 2 months later, he variolated him with real smallpox scab material. The guy did not develop symptoms and seemed to be protected, and that was the beginning of Jenner's paradigm shift, where he said you didn't have to immunize people with the bug that was actually causing the disease, which could be dangerous, but with material from a related disease that was relatively harmless. And frankly, Jenner's discovery, or invention rather, ultimately led to the eradication of smallpox, which was a scourge that had plagued humanity since antiquity.
It's absolutely fascinating, and particularly since this has actually been going on since the 1500s, as you say. But I guess scraping scabs and injecting them doesn't sound so appealing. And we know that vaccines have changed, dramatically since then. So how have vaccines changed?
So first of all, I think it's remarkable to note that when Jenner developed his vaccine, he had no idea that microorganisms cause infectious disease. Nor did he know, of course, anything about our immune system. So the discovery of that microorganisms cause disease really came with Koch and Pasteur in the 18th century. In the 19th century, when Koch in particular learned how to isolate bacteria and grow them in the laboratory. And that's how he understood that these were the things that caused disease and so on. Pasteur also did that. And then Pasteur had a remarkable serendipitous discovery. He found out, without the details, that if you injected animals with either inactivated or killed, rather, forms of the pathogen, or alive but attenuated forms of the pathogen, then that also conferred protection. And so developed these two classes of vaccines, inactivated or killed, or live attenuated vaccines. And it was tried by him for the first time, the latter, live attenuated vaccine, for rabies in a very famous incident. Now, these 19th century vaccines are still in use. So, for example, your influenza vaccine, this seasonal flu shot, is an inactivated or killed virus. It's killed, so it can't do you any harm. And in the 20th century, both of these approaches were used as polio vaccines. And indeed, the live attenuated version of it ultimately took hold, and we have near eradication of polio. So then the 20th century also saw major advances in the life sciences. and associated technologies. So we learned that all proteins are made by going from DNA, which contains information about the proteins, to RNA, to protein. And then people figured out technology called recombinant DNA technology, where you could take a piece of DNA for a given protein, insert it into the DNA into a bacteria, and this bacteria would become a protein manufacturing factory for that particular protein. And so then people said, well, by then they knew about the immune system. So they said, really, what vaccines do is get the immune system activated to target proteins. So why do we have to give any form of the virus or pathogen? We'll just give protein, which we'll make by this recombinant DNA technology. So it'll be safe because there's no pathogen involved, and it will be effective because you'll still end up targeting the proteins. There are some other side issues they had to deal with, but that's the 20th century so-called subunit vaccines, and the HPV vaccine is a perfect example of that. And then to come to our times, in the 21st century, the COVID pandemic made another transformation, because now they say, well, why do we have to give proteins? We'll just give information about these proteins to humans, and their own cells will make the proteins. So that's the J&J and AstraZeneca vaccines, where they just gave cells information about the DNA of the spike protein of SARS-CoV-2, which causes COVID-19. And the Moderna and Pfizer RNA vaccines just gave you the RNA, so your own cells could make the proteins. So vaccine technology, as you can see, has evolved a lot since variolation in 1500.
Thank goodness for that. So, as I've said in the introduction, we now have vaccines for many diseases, but we haven't able to eradicate any of these like we did for smallpox. So why is that? Why is it so hard to eradicate infectious disease?
That's an excellent question. And as you know, smallpox was eradicated by an international cooperative effort led by the WHO, which started in the 1950s. And by 1979, smallpox was eradicated. The last person to get it was a woman called Janet Parker, who got it in a laboratory accident. She was a medical photographer. So why have we succeeded in doing that? One, because of this very organized effort, which of course we could still mount against other things. But there's another reason, and a very important reason. To our knowledge, smallpox does not infect other animals other than us. So once you eradicated it from us, that was game over. So there's this possibility of similar pathogens spilling over by mutations and things like that into humans did not exist. So that was, I think the latter was as important a reason as the first one why we have been able to eradicate smallpox.
And could you briefly describe what you mean by this spillover and what happens when that spills over into animals and back and forth?
When our ancestors learned how to grow crops, their lifestyle changed in important ways. One of the ways that it changed was animals began to be domesticated for farming. So the pool of viruses that circulated in animals and the pool of viruses that circulated in us could be exchanged because of close contact between animals and us. Now, most of the time, an animal virus infects us, no big deal. Nothing can happen because the virus is adapted to the animal, and to cause trouble there, they don't do anything to us. But sometimes, one way that spillovers can happen, and one way alone, is that the same cell in either an animal or a human could get infected by both the animal form of the virus and the human form. And then there could be exchange of genomic bits between the two kinds of viruses. And so now you may have a virus that evolves by chance that is adapted to us and can infect us, but its proteins our immune system or vaccines have never seen before. And so when such a virus starts to circulate, then it has the chance of creating havoc, such as we saw many times through history.
And we call that phenomenon reassortment. Is that correct?
That depends. Yes, reassortment for influenza, it's called reassortment because it happens by swapping pieces of the genome, and the influenza genome really comes in discrete cassettes. So you can swap one out for the other and so on. Yes, reassortment, good word for it.
Okay. So, why do we have some vaccines that give us lifelong immunity, like for measles, but others we need to have annually, like influenza?
So that is the $1 trillion question. If I knew the answer, I don't think I'd be sitting here chatting with you. I'd be using it to make better vaccines. No, I'm joking about that. But no, so I think that's one of the great puzzles that we, the immunology community, does not really know the answer to. And it's not only different pathogens, but different vaccination approaches and technologies also lead to different duration of response. And I think it's one of the grand challenges that we need to confront and overcome in the future.
And there are also diseases we haven't been able to create vaccines for, HIV, for example. Why is that?
Well, HIV is an example, an extreme example, I might note, of a highly mutable pathogen. And many viruses are mutable. HIV is extraordinarily so, and it also replicates quickly in a given person. And mutations happen during replications, so you have many shots at making these mutations in a given individual. But really, we don't have a universal vaccine for the seasonal flu either. So every year we guess which strains might emerge next year. We follow what's happening in Australia to decide what to vaccinate in the Northern Hemisphere and vice versa. And sometimes we guess well and sometimes we don't. But every year we have to design a new vaccine. And as you can see, the situation with SARS-CoV-2 is also becoming similar. So it is this high mutability that presents the challenge because, if you immunize a person with protein components of a particular strain, then the specificity of the immune response will direct a very bespoke response to this strain's proteins. So if you get a new strain, it's a problem, right? So if it's a variant, is a problem. Now, of course, every virus or pathogen has some portions that are conserved across strains because those have to interact with us productively in order to infect us. And we're not evolving that quickly. So the trick is that viruses use very, very many tricks to hide these parts of their genome from the immune system. And the trick is that can we go around that and develop vaccination protocols that might be able to target these conserved bits. And for antibodies that target such things, they're called broadly neutralizing antibodies, or BNAB. And they have been seen isolated from some infected patients in flu, rarely, and also in HIV. But they are usually subdominant responses even in these people, and so on. But their existence is proof that the human immune system can evolve such antibodies. So there's a very big effort with a large community of scientists in both academia and industry that are focused on trying to develop these variant-proof vaccines or universal vaccines. And part of my own research is directed toward addressing questions pertinent to that goal.
What do you think it will take to get there?
I don't know, but I think we are making enormous progress. So I am hopeful that we'll be able to do that. But I am an optimist by nature. But I think there are lots of advances happening, and I think slowly but surely, I hope that we will progress towards doing that, at least for some virus families. And HIV might be the hardest, but we'll see.
So the speed at which vaccines were created for COVID-19 was truly remarkable. What are some of the key lessons learnt from that experience?
Indeed. Operation Warp Speed was a resounding success. And maybe I'll make three observations. So in 2020, the United States government launched a program that was called Operation Warp Speed, whose plan was to accelerate vaccine development against the COVID 19 disease. And it is a remarkable example of how things work and also how well the logistics were handled. But based on that, I would make three observations. There are many more that one could potentially make. But one of them was that it was a remarkable example of partnership between government and the private sector. So the government financially de-risked many things for these private companies that were developing these vaccines so they could do many steps in parallel, pursue many steps in parallel that they would normally pursue sequentially to make sure that their investment wouldn't go awry. But the government de-risked much of that. So I think one lesson to take from this is that there are ways to have productive private-public partnerships. The second thing I might note, and this is very important to note, that the vaccines that first came out were the ones from J&J and AstraZeneca, which I told you were based on these inserting DNA into and non-replicating virus called the ad vector, and the RNA vaccines, which Moderna and Pfizer BioNTech, and they inserted the RNA. But let me say that research on both of those technologies for vaccines had been going on for 10 years. It's not that they appeared for COVID-19. They were there and under development for a long time. And the COVID-19 crisis allowed them to be deployed and tried. And so the lesson I would take from that, going back to what I was telling you a little while ago about variant-proof vaccines, is that I think we should keep making such investments in cutting-edge technologies that may or may not pan out because, it'll help us prepare for the future. And a third observation is that perhaps we need, as a global system of countries, to invest more in biomanufacturing capabilities in diverse parts of the world, because that may improve some of the access issues that we did encounter. That is, access was not uniform across the very countries with various types of economic strength and development. So those are three observations I might leave you with.
Three excellent observations. Operation Warp Speed was brought about in a time of crisis. Why can't we have Operation Warp Speed in a time of peacetime so we are ready for the next pandemic?
So this is a question really not so much for me, but this is a question for policymakers, because they make these decisions. But I really do think that you're right, and that there should be serious thinking about what can we do at normal times. I mean, we can't do what we did for Operation Warp Speed. I mean, you can't tell companies develop whatever you want and it's de-risked. But what is the right balance is something to explore.
So we touched on universal vaccines being one of the technologies of the future. Are there any other technologies we can expect?
Well, that's always hard to predict, and I won't do that. But let me just say this. Historically, what we have seen is, over time, vaccination procedures have become more effective in protecting us, easier to administer, and progressively more safe. And I expect that those trends will continue. And as I mentioned, it is my hope that we will, at least for some classes of pathogens, learn how to create variant-proof vaccines, but also learn how they will lead to longer duration of memory, as we talked about a little while ago, and also protect surfaces that are exposed to these pathogens rather than just blood alone. And all of those things happen sometimes in regular in vaccines we take, but sometimes not. And so, can we truly understand how to do that repeatably across? So, like I said, safe, more efficient. I expect that to happen. Hopefully, more variant-proof vaccines, and hopefully ones that are more durable.
I hope you're correct. So, despite the positive impacts of vaccines on public health, there's still opposition to vaccines. What are some of the key factors, do you think, that are driving vaccine hesitancy?
Well, vaccine hesitancy and opposition is, first of all, not new. And I think it's perfectly understandable. After all, you're asking a healthy person to do something that seems scary. You're asking the person to take some component that is related to a dangerous pathogen and inject it into yourself. I think it's natural for people to be concerned and scared about that. I think what is required in order to try and address this concern, very real concern, is to lay out very, very transparently and with everything that we know what are the benefits to the individual and to society, especially the more vulnerable. That is, by my taking a vaccine, a more vulnerable person is also protected if a lot of people take it. And after describing those benefits, also be very transparent about what is known, at that time, what is known about the potential risks to any given individual. And there are risks to some individuals. And I think only by, and perhaps also point to historical record of the Vaccine Adverse Event Reporting Agency, where they keep track of existing vaccines, how many adverse events have been reported, and thereby get a sense of statistically how likely is it that an individual will be at risk. I think if we lay out all those facts very clearly and also debunk some myths that are also around, by being transparent, being open and honest and so on about the risks as well as the benefits, hopefully that will cause more people to decide that it is worth the risk.
Do you think that we're not already doing that, though, or do you think that it's just a case of we could be doing more?
I don't. Now it's only personal opinion here.
Yes, of course,
Okay?
of course.
This is not fact, but I can't. I feel that the messaging was, again, I speak for where I was, but I think while the messaging was very clear to those who perhaps were not more likely to be hesitant, perhaps the messaging was not as clear to those who are more likely to be hesitant. And also, I don't know what to do about this, but there was a lot of misinformation being also peddled. And I'm not the expert to say how to stop that.
No, and you're right. It is very different in in every corner of the globe. And as I said, it changes, dramatically, depending on the day, let alone when being in a time of crisis. So, which can give rise to all different misconceptions, which we saw during COVID.
But the fear that you'll get autism if you're vaccinated for childhood diseases is largely the product of one paper that was retracted. So how do you control that is a challenging thing. But I think we also should lay out the risks very, very clearly because there are risks.
Yeah. But even though that paper was retracted, that's still, a relatively wide held belief in some places.
Absolutely, which periodically causes measles outbreaks.
Yes, which we're seeing globally. Changing pace a little bit, what can we expect from your Miegunyah Distinguished Fellowship Public Lecture on the 25th of March here in Melbourne?
Well, first of all, it's a great honor to have an opportunity to give this lecture. Look, I'm not a professional historian by any means, but what the audience will hear is the reflections and observations of a scientist on how vaccination procedures have evolved through the centuries with more detail than what we did during this short chat. And I will try in the end to give a very high-level flavor of the kinds of things that people are trying to do to work towards these variant-proof vaccines. Of course, it will be a tiny sliver of all the efforts that are ongoing. So that's roughly what I'll try to cover.
Well, we're very much looking forward to hosting you here in Melbourne. And finally, you have had a truly fascinating career. As an engineer working in immunology, what unique perspective does an engineering approach bring to the study of immunology?
Yeah, indeed, fascinating is an interesting word for it. Perhaps you should have said that don't try it at home or something like that. But
you don't recommend it?
No, I do recommend it, but it requires a leap. For me, it required a leap of faith because my formal biology education in a classroom ended in the 8th grade. So I was 38 years old when I made a big change in field. But basically, I think as an engineer, some kind of a cross between an engineer and a physicist, now several are doing what I do, but I think that whenever people from another field bother to learn about another field, and that's very important, that you have to learn about that field, and you have to work with people who are card-carrying people in that field. I mean, my lab could not have done anything without of any usefulness without my brilliant students and postdocs, of course, but also my collaborators who were all card-carrying immunologists or virologists. But when people from these different disciplines are brought together, sometimes you can see the same problem in a completely different light, in a new way. Just to give you an example, as an engineer or physical scientist, or any other engineer or physical scientist, many of them know how to think about many different components acting together cooperatively to give you a coherent final answer. So this thinking about systems and thinking about collective effects is something that I think engineers and physical scientists can bring to the table, which often can lead to interesting give and take, which leads to a new way of thinking about something. And of course now computation, which I do a lot of, but now even more with AI and ML, will, I think, together with the kinds of things we do with physics-based computations, machine learning, and so on, that'll add another dimension to all of life sciences, and immunology is not going to be an exception.
So what advice might you give to, young budding engineers or immunologists or physicists?
I'll tell you exactly what I tell them when they ask these questions whenever I'm the student invited speaker somewhere. And I say that, well, do what your heart says is right, even if everybody and your passion is about that, because everybody else will initially tell you that this would be a very bad move and a very dangerous move and risky move, and probably is. It probably is. And but if you'll never be a contributor in any field that you're not passionate about. So if you feel that you want to work on a new field, you should do it, and keep an open mind.
Well, I think that's a great place to end on. Arup, thank you so much for your time today. It's been absolutely fascinating.
Thanks. I enjoyed it very much. And I look forward to seeing you in Melbourne in a couple of weeks.
Yes, can't wait. Really looking forward to it. And thanks to all of you for listening to Infection and Immunity Evidence Explained, a Doherty Institute podcast. Listeners are warmly invited to attend Arup's Miegunyah Distinguished Fellowship Public Lecture titled Vaccination Through the Ages, hosted by the Doherty Institute and the University of Melbourne's Faculty of Engineering and Information Technology on Monday, the 25th of March. To register, please visit our website, doherty.edu.au. That's doherty.edu.au. If there is a specific topic you would like us to cover, please get in touch with us by emailing doherty-media@unimelb.edu.au. That's doherty-media@unimelb.edu.au. If you've enjoyed our podcast, please rate and review on your favourite podcasting app and subscribe if you haven't already. The Doherty Institute is a joint venture between the University of Melbourne and the Royal Melbourne Hospital. See you next time.